Thank you, Suzanne, for that introduction. Like you said, my name is Jack Fetters. I'm a conservation biologist for the Xerces Society for Invertebrate Conservation. I'm here with my colleague, Emily Blevins, who is a senior conservation biologist for the Xerces Society. Today, we're going to be talking about the weird and wonderful lives of Oregon's native freshwater mussels. So before I dive in, I kind of wanted to give you all some background about our organization, the Xerces Society. We're a nonprofit dedicated to invertebrate conservation, as we were established in the 1970s. As a science-based organization, we both conduct our own research and rely upon most up-to-date information to guide our conservation work. If some of you have heard of Xerces in the past, they tend to hear about more of our work with bumblebees and butterflies. But in the 2000s, Xerces developed a freshwater mussel program that dedicated to work toward a future where water bodies contain healthy, diverse, connected, and abundant freshwater mussels. And so that's what I'm going to be talking to you today is some of our work with freshwater mussels and a little bit about them. So to kind of bring it back to a bigger picture here first, freshwater mussels belong to the phylum Mollusca, um, which makes up roughly 1/4 of the species we know on Earth. Here we have some examples of mollusk, including an octopus, a snail, slug, marine clams, pea clams, and the giant clam. Um, all of these mussels' life cycles can vary, but most have have a larval and adult stage, but some undergo direct development. Um, Also, the mantle is what ties these species together. Even if this doesn't result in a shell in all mollusks, the mantle is a layer of tissue that creates the shell for mollusks. And like I said, all mollusks have this, but some don't create a shell with it. Furthermore, there are other mollusks that are more closely related to freshwater mussels that are considered non-native invasive species. Kind of zeroing in here a little bit, wanted to explain the difference between non-native mussels and our native mussels here in Oregon. The first two examples that I have here are the Asian clam and zebra mussels. They are different from freshwater mussels for a few reasons. The first reason is our native freshwater mussels require a host fish to complete their life cycle. I'll give some more details about their life cycle and the host fish relationship here in a few minutes. But the second reason, how they're similar or how they are different, is native mussels have a burrowing foot, a fleshy foot used to anchor themselves into the substrate for benthic burrowing. They also have two siphons or apertures. One takes in water and filters it for feeding, and then one lets out water. Um, some other non-native species, like the quagga mussel, have their ability, rather than burrowing, is attaching themselves in other forms rather than the burrowing foot. Generally, non-native species live about 7 to 9 years and can sometimes reproduce multiple times a year, while as some species of freshwater mussels can live to 100 years. Yeah, so sometimes these non-natives can be competitive with the native species of freshwater mussels. And in certain conditions, they require our native mussels require some specific habitat needs. And we've kind of broken these down into 4 groups of habitat needs, and the first being perennial flows with minimal drought conditions, otherwise, consistent, constant flow. They can't be out of water for long periods of time. It's lethal to them. And also on top of that, they require stable habitat features such as rooted vegetation or boulders. They like to anchor themselves around these things because it's consistent, and they generally don't move throughout their entire lives. Um, and they also require burrowing material within these stable habitat features, such as gravel and fine sands that allow them to anchor themselves with that fleshy foot that I spoke about earlier. And then what's really unique about freshwater mussels is they require a host fish in order to complete their life cycle. These animals being very sedentary, they using a host fish to complete their life cycle, which again I'll give some more details about in a few minutes, is their only way to move around in a linear system of river or stream. And so moving forward with that, some areas where you can find freshwater mussels are typically in lakes and ponds, streams and creeks, large and small rivers, and perennial water bodies. Uh, some important things to know about where you can and can't find freshwater mussels is you can sometimes find them at really high elevations. They've been reported at elevations as high as 10,000 feet in meadows. Um, they can't handle brackish water. Um, they can they can survive in freshwater tidal areas in like the lower Columbia, which is unique to the Pacific Northwest. And they can also survive in interdunal lakes on the West Coast. So earlier I kept mentioning this fascinating reproductive life cycle that freshwater mussels do. Well, here we have a great figure developed by Monica Blanchard from WDFW. So essentially what happens is a male freshwater mussel will release their sperm into the water column, and a female takes in the sperm to fertilize her eggs, which then develop into a larvae known as glaucidia. These glaucidia are released in packets that resemble um, a, a bite-sized, I'm sorry, these packets are released in conglutinates, which are these like bite-sized packets that host fish are attracted to. Once a host fish takes a bite of this so-called food, aka Glaucidia packets, the Glaucidia latch themselves onto the host fish and gills and scales. They remain on the host fish for several weeks and then drop off into the substrate and develop into adults. So some really cool, interesting stuff going here, really unique life cycle to these freshwater mussels. And like I mentioned, this process is how they're able to move up and down linear systems of a river or stream. And when they fall off, it's really a lottery of whether or not they'll survive because it's dependent on where the fish goes and where they drop off. So hopefully they fall off in the right place at the right time with the right conditions, like I mentioned before. So in addition to the Glaucidia packets that our native freshwater mussels in the Pacific Northwest can do, there are some species in the eastern US that have more uh, intricate ways of attracting the host fish. And they use things, they call them lures or egg mimics and to attract them in. And I have some videos here that I'd like to share with you about of that going on that are pretty fun. So these videos were created by Chris Barnhart from Missouri State University. So what we're seeing is that's a mussel displaying its lure and a bass taking a bite out of it and getting infested with its glaucidia. And as soon as they get in there, they latch themselves on to the gills and the scales of the fish, and they hang on and incite themselves into the gills and feeding off them. And they hang on for, like I said, for about several weeks, growing and developing into adults. So this is a different species using a somewhat different method than the lure of attracting the fish. They will actually capture the fish and infest it with their glaucidia, and they'll hold on to it for a little bit, and they're just and continuing to infest it with the Glaucidia, getting in its gills and scales, like I said before. And during this, it may seem like it, but the fish typically come out unharmed, just kind of go through a little bit of a whirlwind. Um, but generally they come out unharmed and totally fine. Eventually they will let them go and they just go off to do their thing. And so this one is its method of attracting a host fish is the most similar to our species here in the Pacific Northwest, where they're releasing these packets that you're seeing come out of it, and the fish come up and nibble and think they're getting a tasty meal, but in fact they're getting infested with some Glaucidia. Yeah, so moving on from that, I want to tell you more specifically about the species of freshwater mussels that we have here in the Pacific Northwest and in Oregon. Oh, went too far. There we go. So there are three genera that we have here. The first being the Western pearl shell. And like I mentioned before, they have these siphons or apertures used to one for taking in water and one for exhaling water. And they use these filters and what's called papillae to filter out things and feed on things in the water column. So this particular species, Western pearl shell, they can live up to 100 plus years old. They require salmonid, or so trout, for their host, for their as a host specialist. They're the wildest western or the widest western North American distribution. And although they're widely distributed, sometimes they can be locally rare. But when they are abundant, they can exceed beds higher than 10,000 individuals, all located within one place. And they can be ID'd by the color of their outer shell and the papillae arrangement on these apertures that I've been talking about. So the next group is floaters, Anodonta species, and there's multiple different species within this group. However, although they look very similar, they are genetically distinct. And this group of mussels can live to be 10 to 20 years old. Generally, they're considered a host fish generalist. They have a wide distribution as well. And like I said, the species within this group have similar appearance, but generally distinct or genetically distinct. And they can also be ID'd by their papillae arrangement as well. You can kind of see the difference in this one compared to the Western ridge. And the last one is the Western pearl. I apologize, Western pearl. But the last one is the Western ridge. And it has a lifespan of 30 to 60 years. They typically use sculpin as a host fish, sometimes not, but that's what they seem to typically use. They're more limited in North America distribution. Recently, there's been some research that have determined a decrease in 43% of their populations throughout the PNW. They're considered more cryptic. They can be a little bit more difficult to find. They really like to hang around those boulders that I mentioned earlier. And like I said, they've experienced declines, and they also can be ID'd by their papillae and also by their shell shape. A little hard to tell in this photo, but they do have a distinct ridge when you're handling them outside of the water. And then with that, I'm going to hand it over to my colleague Emily, and she's going to tell you a little bit more about what Xerces does with the Luckiamute Watershed Council.
Thanks, Jack. Yeah, so Xerces, as Jack mentioned, has had a freshwater mussel program for a number of years. Part of our early work was to really help better understand the distribution of freshwater mussels across their ranges. We had some information, but it's often very scattered. Some historical records. Of course, the tribes have a long history with freshwater mussels. We'll talk about that. But in general, we didn't have a very good understanding of historic distribution of mussels versus current distribution to understand their conservation status. So one of the projects that we've undertaken has been to try to conduct more surveys and to gather more information about both the distribution and the status of mussels. And this is where partnerships have been really critical. With the Luckiamute, we've had the good fortune to conduct surveys at various properties, be able to access areas that we had no prior information, and really be able to better expand our understanding, particularly in some of these watersheds that really had been under-surveyed until more recently. So it was really fun to kind of pull together this map of the Luckiamute Basin and be able to look at some of these locations. Really, we only have had recent records from our collaboration with the Watershed Council. So it's been really wonderful to be able to start to fill in those gaps. As you can see, there are only a handful of locations where we have documented the different species. So you can see the blue pins are the floaters. Western pearl shell are those black hexagons. We don't actually have any records of the western ridge mussel in the basin, but that is another species that we know occurs in some of the Willamette River tributaries and in the main stem. And so there's still so much to learn about this. And so we can talk a little bit about surveying for mussels, but it's been a really wonderful opportunity to collaborate to really expand what we know, because in many cases these animals occur and simply we haven't looked for them, we haven't been aware of them, we haven't appreciated them. So it's been wonderful to partner. Next slide. So as part of that, in 2018, Xerces received a grant from OWEB to assist watershed councils, agencies, and others to conduct surveys for mussels, specifically in advance of habitat restoration efforts for fish. And that's in part because mussels can be incredibly sensitive to abrupt changes in streams. As Jack mentioned, they really are unable to handle being dry. And so when we dewater streams, even temporarily to do construction work, even if the outcome is beneficial, even that short period can really impact mussels. So as part of this project, we have collaborated to visit a number of creeks just to determine if mussels are present, and if they are present, what we can do to limit impacts to them as we undertake these restoration projects. So it was really interesting to visit a number of sites that had been slated for restoration activities. And in some we found 0 mussels, and in others we might find 2. In one we found a larger number that we were able to then work with a contractor to avoid impact. So it's always interesting. It's very difficult to tell in advance if mussels are going to be present at a site and which species and how many. So this is a photo I took at Vincent Creek with a western pearl shell on the right and a floater mussel on the left, and then a lot of native snails as well. We've also benefited from having Luckiamute Watershed Council staff on the lookout and sharing their own observations when they're in the field. So thank you. And as far as how we look for mussels, so as we've said, mussels can be really cryptic. You've seen these pictures, they look like rocks. So it can be really easy to overlook them unless you're out there with your face in the water, really hunting for them. And so we can do that a number of ways, whether it's by snorkeling or using something like an aquascope, which is basically like a snorkel mask on a tube. And that's allowed us to really investigate some of these areas. And so, for example, in the image at left, we're looking at a western pearl shell bed that is close to shore, so you could see with an aquascope, but great view if you're able to snorkel. So if you haven't tried snorkeling in rivers, we highly recommend it. It's a really great way to see fish. It's a great way to see mussels and really appreciate what's under the surface. Next slide. Okay, so here's the test. You don't actually have to tell us, but in your head, can you pick out the mussels and how many do you think there are? We promise they are here. And if you become an official snorkeler, you'll get much better at this. It takes a little practice. But mussels really can be quite cryptic in part because they're the same shape and size as rocks, the same color. They might be covered by algae, which is what you see in this image. All right, Jack, should we help him out? Okay, Jack spotted at least 11. We're debating whether there could be a few more in this photo, but oftentimes when you spot mussels, what you're seeing are those apertures that Jack was pointing out, because their mantle tissue, which forms the edges there, is usually a slightly different color. You can actually see into a mussel when they're open and filtrate. You can see their gills. It's pretty incredible. They're really a fundamental part of the river, totally open to the flow. Next slide. And so freshwater mussels, we care about them because they're important in their own right, but they really are incredibly fundamental to our aquatic ecosystems. And it's really been underappreciated how much they contribute. So, because they filter to remove food from the water column, to actually breathe, to reproduce, all of those activities are important for them, but as a byproduct of their filtering, they're actually doing a number of things to improve water quality. So they feed on algae and bacteria, so they can improve that clarity of the water as well as the health of it. For example, mussels can filter out and actually feed on E. coli bacteria, which can make us sick. They can remove pollutants from the water column, including things like pharmaceuticals or personal care products, flame retardants, things like that. So they're actually pulling that out of the water. They also create habitat. So we think of them as animals, but when they occur in a bed of 10,000, they're really fundamentally a part of the environment. They create almost a reef-like habitat that is really valuable for other species. So they can serve as habitat for snails. They grow food on their shells, essentially. So it's like a buffet. Other animals will live inside them, like crayfish inside of this empty Western Ridge mussel shell. So they really can change the environment that they're in. They also do serve as food for a number of species. So this is a really neat photo. We have a lot of mammalian predators like to snack on mussels. So muskrat, raccoon, and otter all like to eat mussels. And to feed on them, they have a number of techniques, but one is often to bite the shell. So you can actually see a tooth hole in that image. With their little paws, they'll rip the other side open, and then they'll eat the animal inside. And this was a great photo because you can actually see the evidence, that footprint of the predator that fed on this floater. So they're important for the food web, and particularly for as part of the nutrient cycling that they do, they can capture food from the water column and anything that they're not actually going to eat. Rather than go through their digestive system, they kind of package it up in mucus and spit it back out. And that's food for grazing aquatic insects. So there's research that shows that you get a greater abundance of other aquatic macroinvertebrates around mussel beds because they're really cycling this and concentrating this food source. And so that's great for fish because that means that they're providing a really concentrated resource for fish as well. And mussels do a number of other things. We could talk all day about how great mussels are, so we'll keep going. But yeah, they provide really fundamental ecosystem services for us. And this is just a diagram to kind of depict that, how they're filtering 24 hours a day, nearly continuously through the year. They live potentially 100 years. They're maybe tens of thousands of them. So, the overall impact that mussels can have on the environment can really be quite large. But if we are not measuring it, we're not appreciating it. So, they're incredibly important animals. And this is a really great example of how that filtration that mussels are doing, for their own good really has this beneficial impact. So these two tanks both have river water that's fairly turbid. You can see that the tank on the left has mussels, the tank on the right doesn't. And after 30 minutes, this is the difference in water clarity you get with freshwater mussels. So they're really, their capacity to change their environment is quite large. Next slide. And so they've been really culturally important in the Columbia Plateau and Pacific Northwest region. We have examples of shell middens, which is the image at left. These are essentially areas where there's shell material left over after mussels were harvested for food or other uses. We have examples of jewelry that was created from punching out Western pearl shell shells. So they've been important as a first food. They've been important for trade and cultural use. And so, the mussels really have this impact on human life that we really should appreciate more. So what's really cool about freshwater mussels, if you don't already think they're cool, is that North America is a biodiversity hotspot. We have really close to a third of all freshwater mussel species found on Earth, just in North America. Most of that diversity does occur east of the Rockies, and particularly in the southeastern US, because as you saw, Jack walked us through the native species out west. So we have fewer species out west, but we are part of this really incredibly diverse fauna in North America. Unfortunately, freshwater mussels, as well as freshwater snails, are the most imperiled wildlife that we have. And most people are surprised to hear that. We think of our declining fish species, we think of birds, we think of, perhaps the monarch butterfly, but freshwater mussels and snails really are going extinct at the fastest rate of any of our species. There are more than 90 mussels that are already federally listed as endangered or threatened, and more than 30 species have already gone extinct. And so it's really a conservation crisis for this species group. And we can all pinpoint some of those reasons. There has been a long history of change in our rivers, whether increased pollution, dam building, straightening of channels, removal of wood, dredging. We use our rivers pretty heavily and have for hundreds of years now. But mussels are also facing a lot of challenges in a really fast changing environment. So these are all images from Pacific Northwest ecosystems, including the South Fork John Day River, which was one of our mussel study sites that's during our surveys went dry and killed mussels that were present there. Another site in the middle there is near Lake Tahoe, where a mussel relocation site burned incredibly deeply several years ago. And then when we have debris flows or decreased oxygen in rivers, it kills fish. But the example here is from the Klamath River following the McKinney fire, and that was also a site that had freshwater mussels. So these mussels are facing a lot of historic and emerging challenges that as a society, we have the potential to really make a difference. Next slide. So it's a huge challenge. Xerces has developed this Western freshwater mussel conservation program because we believe that there's a lot that we can do to ensure that our species have a much better future. As Jack mentioned, our goal with the program is to envision a future where our water bodies have abundant, thriving, healthy mussel populations. And so to ensure that we're doing our part for that, we do a number of different things, including conducting surveys, like I mentioned. We assist with mussel salvages. So where mussels could be impacted by human activity, we try to assist in those instances to get them out of harm's way. We do trainings to help educate people about mussels, including many of the biologists who are working in these streams already and could also work on freshwater mussel conservation as well. And then we also develop and provide a lot of resources and conduct research to make sure that the best information is available out there, and so that folks know about mussels and their importance. Next slide. So there are also things that, the average person can do to help freshwater mussels. You don't have to be a mussel biologist to do something that really makes a difference. Much of the activities of things like watershed councils goes a really long way to improving conditions for mussels. So protecting and restoring stream banks and river bottoms is really critical. Mussels live in those places, and as Jack said, they don't move much during their life. So if you've got a mussel bed somewhere, helping it thrive can mean making sure that those good places stay intact, and those places that need help, we are targeting them. So habitat restoration can really improve conditions for them when it's protective of them as well. Clean water really is critical for mussels. They do have that capacity to remove pollutants from the water, but they also can be really impacted by having so many different challenges at once. And mussels are also unique in that they can be some of the most sensitive animals to certain pollutants of any that have been tested. So a chemical like ammonia, which might come out of a sewage treatment plant, is highly toxic to freshwater mussels. And so, investing in infrastructure and resources to ensure that our water stays cleaner will ensure that they can add that added benefit to water quality without being really heavily burdened. As we said, freshwater mussels, they really, they're dealing with these situations where our rivers and creeks are running dry due to drought conditions and water use. And so anything that we can do to reduce water use really has a huge impact on these rivers and therefore on our mussel populations. So we absolutely recommend, you're here, so obviously you support a watershed council, but groups that work to really improve and advocate for our wild spaces, as well as our inhabited spaces, are really critical to mussel conservation. So now that you're so excited about mussels that you can't wait to go look for them, there are a couple things to consider. So in Oregon, as well as in several other states, mussels are protected by the state just to ensure that declining populations are not further impacted by humans. And so to handle freshwater mussels in the state, unless you have a reserved treaty right as a tribe, you do need a permit to handle live mussels. So we recommend that if you're interested in mussels, to go out and look for them, but don't pick them up, don't touch, leave them where they are. But you can get some great photos of them. They don't run away from you, they don't swim away, so very easy to get a good look. You can pick up shells and you can keep them. So we have a shell collection that we carry around to share with others. And it's also a great way to identify the species. As Jack was saying, you can identify mussels, both living from, what those papillae look like, but also from their shell shape and some of the features on their shells. And so if you are a fan of iNaturalist, you can take photos of shells you might find, or underwater photos of mussels if you've got a cool camera. And post them to iNaturalist. And that data actually does come to Xerces, where we go and we gather that information to help us improve the maps that you see. So, so you can be a really active participant in mussel conservation by assisting with helping us find where they are. I do have this picture up. People do occasionally harvest mussels, and one of the main questions we get is, can you eat them? Because of course, we showed an example of a shell midden and talked about their importance as a first food. You should keep in mind, first of all, that you do have to have a permit, so it's not permitted to just collect them for food. But my favorite line from this review of someone who posted online that they had tried to cook these Western pearl shell collected from the Willamette, that I think the quote was, no matter how much salt and butter they added to the mussels, they still tasted like dirt. So, these are animals that live in the muddy bottom of the river. They're not in that briny, delicious soup that a coastal marine mussel is in. And so just definitely recommend not eating them. Also because we do have many more pollutants in our rivers now than ever before, and they are potentially being concentrated in mussels. So look, don't touch, don't eat, but really we can appreciate them for what they're doing. But so with that, I know Jack and I would really like to thank the many agencies and organizations and individuals who support our work. We can't do this alone. Mussels don't live in one place, and we can't track them all. So our ability to work with the public and to work with organizations like the Luckiamute is really valuable. So we want to say thank you to all of our funders and collaborators and supporters. And with that, next slide. I will just put in a plug that Xerces is a donor and member supported society. And so if you're interested in any of our resources, you can visit xerces.org. We've got a lot of information there about many invertebrates that we work on. And if you're interested, you can donate to help support the work that we do. And I like to just finish presentations with this really awesome picture. You know, I talked about some kind of depressing things, but there are still abundant, beautiful, you know, freshwater mussel beds out there. This is a Western pearl shell bed from a stream, actually in Washington. But this site had an estimated 25,000 Western pearl shell in, you know, the size of a backyard above ground swimming pool. A very small area, but just, you know, this thriving, really cool place. And so it's a good reminder when we visit these places what's possible. So with that, I think that's our presentation, but I know would be happy to answer any questions.
So the question in the Q&A is, are there historical population counts for coastal populations? And if so, are there any insights or studies on the impact of broadleaf spraying like glyphosate over the last 60 years throughout the coastal range?
Yeah, I could take this one. So we don't really have many historical counts for mussels really anywhere. We're only recently developing, you know, plans and tools for being able to resurvey or conduct more precise surveys of mussels. And so we do have some historical observations, and the tribes also have, you know, ample evidence of far more abundant mussel populations historically in various places than there are now. In the Coast Range, I know that there was a study published that reported observations from locals about the loss of a really abundant mussel bed in the Siletz River. But otherwise, we don't really have good information about historical abundance. We are gathering a lot more information about distribution of mussels currently in the Coast Range as part of ODFW's Aquatic Inventories project. They do surveys for salmonids, but as part of that, they report on freshwater mussel observations as well. And so that's not abundance information, but it is distribution information. And there was a really neat article, research article that was published that analyzed that data. So that's a neat resource. So hopefully moving forward, we'll know more. Regarding glyphosate and mussels, it's an interesting question. As I mentioned, mussels are often the most sensitive species to certain pollutants of those that have been tested. And glyphosate's an interesting one because of the limited study that I'm aware of. Glyphosate itself has not been shown to really have an impact, but some of the things mixed in with glyphosate can have an impact. So we often test the toxicity of chemicals on mussels using their most sensitive life stage, which is Glochidia or juveniles immediately that have, you know, just fallen off of the fish. That's when they really have kind of the least protection from the environment. And in those cases, the test showed, I think, that a surfactant that was added to the glyphosate to help it stick to plants was actually somewhat toxic to those young life stages. And then also a salt, I think that was an additive to the glyphosate. So So yeah, certainly we don't know everything about how those chemicals are affecting them, but some limited study has shown that some of those additives can be a problem for them. But we don't know what effect that's had on populations as a whole.
Great, thank you. How difficult is it to reintroduce mussels into a stream?
Oh, I'll kick this one to Jack, who has a background in mussel propagation from back east.
Yeah, so I worked at a mussel propagation facility in Tennessee for a little while, and we would use that as a tool to reintroduce populations or do some population augmentation. And it it can be a tough tool. I think it's a very useful tool. Um, Essentially, the ways that I've seen it used is if you have like a population in a downstream area that's doing well and abundant, and then maybe in the upstream areas they're not doing so well. They may take some adults from that abundant population, um, grow them out in a facility and produce some juveniles and grow them out until they're past that stage that Emily was talking about when they're juveniles and they're extremely susceptible to pollutants and toxins. And then they release them in those areas where they may have occurred in the past but no longer do. And that can kind of, give the population that used to exist there a bump and, the ability to um, grow and resettle themselves.
Very cool.
Yeah, I hope that answers your question.
It answered mine. That was great. So clear cutting being a pretty common practice in the Coast Range, does it affect mussels? And if so, how?
I can take that one. Yeah, the effect of an activity like clear cutting, it's definitely really, it depends on how that's affecting the nearby stream. So we know that in many places, increased water temperatures are affecting mussels, and in some portions of their range, they're already really experiencing kind of their thermal limits. So that can be a challenge. Our Western mussels, we don't know their temperature tolerances. I think we are starting to have some research done on that for certain species, but we don't really know kind of what they're able to tolerate at this point. Other impacts from the clear cutting, if there are things like landslides or debris flows, we know that buried mussels can really struggle to kind of burrow up through some of that sediment and be able to breathe. It's very possible to suffocate a mussel. And so impacts to the actual stream itself from something like a debris flow following clear cutting could certainly kill off mussel beds. We know that of at least one mussel bed that was being studied in California, when a fire came through, a subsequent debris flow did eliminate that mussel population. As far as other impacts, mussels really need that riparian vegetation to help stabilize the banks. They are often found in bank habitat. And so if you also lose that riparian vegetation, that can really impact mussels. When you see those, straight eroded banks, that's habitat that mussels really can't be found in because they struggle to stay in place against the flow of water. And so there's a lot of ways that clear cutting could impact mussels. But yeah, I think more research would be really beneficial. I know there's been a lot of research into things like the effects on aquatic insects or fish, of course, amphibians, things like that. But we don't have those studies for mussels. So we really need to invest in that kind of research.
Great. Has any mixing or hybridization between species been observed as of yet?
That's a good question. I'm not aware of that in our Western mussel species. They are highly divergent genetically. So, for example, the Western ridge mussel is the only member of its genus on Earth. And so it's quite different than the Western pearl shell. Yeah, Jack, are you familiar with any examples of that from Eastern mussels?
No, and like as Emily mentioned, they're highly diverse in the eastern part of the U.S. But I haven't heard any accounts of hybridization going on myself either. It's still, I think, like similar to the floaters. Some of them are very similar physically, but genetically very different. So I think in a lot of scenarios they may not even know, just because it's hard to tell who's who, just based on looking at them. So, but I haven't heard anything specifically of that going on.
Yeah, that would be really interesting, because you could imagine if you have a highly diverse set of species and everything's just in the water at the same time, that you could definitely see. But yeah, I haven't heard of that. That's a good question.
So right now we don't have any questions in the queue, so it's my turn. My first question is, have there been any partner studies with like archaeologists to study midden beds to kind of do historical population counts that way?
Yeah, so the Confederated Tribes of the Umatilla Indian Reservation is a really they have an active freshwater mussel research and restoration project. They have archaeological sites that they have excavated for freshwater mussels. So they know they were a really valuable part of their tribe's diet for a very long time. If you go to their museum in Mission, Oregon, I highly recommend it. They have a display about their use of freshwater mussels, and they also have a really amazing archive of freshwater mussel shells. And so they've used that information. I don't think to this is a good question. I don't think to be able to determine exact numbers or past abundances, but they were able to compare species composition in their diet. So they relied much more heavily on Western pearl shell than on floaters or Western ridged historically. So that's pretty interesting. I think they probably have some additional research that they've done that I might just not be aware of. But yeah, isn't that really cool that you could excavate through and really see that historical value of mussels? Yeah.
Oh, cool. Thank you. And since, come on, guys, you gotta ask more questions, or else it's gonna be another weird one for me.
I have one too.
Oh, go for it, Suzanne.
Okay. Yeah. I am wondering, you mentioned that some of these species are host specific and some are generalists, and I don't remember which is which, but I'm wondering if it's a case where like biologically they simply, like if salmon and trout, for instance, were no longer in a stream reach because of a dam or a blockage, would the mussel also then have no way of reproducing or dispersing, or can they utilize other species if need be?
Jack, do you want to take a?
Sure. Yeah. Especially for those host-specific species like the Western pearl shell, which uses salmonids, they would eventually die out. There's some research that shows that they can use some other ones, but it's not nearly as effective as using salmonids. So they would eventually die out. Now, the one that you're thinking of that's more of a generalist, the floaters, they like you said, they're more of a generalist. They tend to hone in on, from what I understand, is cyprinids, so like minnows, type fish, and so hard to say with them. They might be able to figure it out, but I would say Probably not. It would depend probably on the fish that was, to be extirpated. So yeah.
Okay, thanks.
Next question. Why was the Siletz River population lost, which I think means the mussel bed that was.
Yeah, I was going to pull up that study briefly because it was really just there was someone who was studying Western pearl shell and their use of salmonids and looking at the effects on fish, which, we should mention just briefly that freshwater mussels, they are parasites of fish. So when you saw those Glaucidia attached to the mussel, they actually are benefiting. They're getting like food from fish blood when they do that, but they the impact to them is so minimal. Often very small number of Glaucidia attach and are successful. The fish essentially treats it as though it's like a scrape, it's a very minimal impact to them. And then the mussel is falling off pretty shortly after. So this study that mentioned that about the Siletz was looking at how the fish handled having the mussels attached. And the study is from 1978. So I'm just glancing through really quickly to remind myself of what it was that they noticed. But I think they just kind of casually mentioned that they saw a decline. I'm not seeing it quickly, but often, the challenge for us when we see these changes in abundance or even just presence of mussels at a site, we really don't ever get to find the answer. In some cases, we will simply find, a bunch of shells and we're kind of left to wonder, did this happen over a long period of time? Did these mussels all die at once? It leaves evidence behind, but it's so hard to interpret how quickly those things happened or exactly when, unfortunately.
Okay. How much is known about places where the population has many older individuals but very little recruitment of juveniles, and the processes or causes involved in this phenomenon?
Yeah, Jack, I don't know if you want to mention some of the interesting studies about water temperatures back east or host fish. I could tackle it.
Yeah, sure, if you'd like to tackle it.
Sure. Yeah. There are some really like kind of cool examples about how sometimes we're just the problem and we can fix it if we only cared or knew. So there was one example, I think it was out of Missouri, but there was a population of mussels below a dam that was not reproducing. And they determined that the water coming from the dam was just too cold, and so they ended up mixing the water to bring it back to a much more normal temperature. And they actually saw the mussels start reproducing after they did that. So another example is a species of mussel that's found in the northeastern U.S. And essentially the mussels were above a fish barrier, and when they started moving their host fish above that fish barrier, they were actually able to start reproducing because, well, they had been, trying, but there was no fish there. So some really nice examples where, the issue and the solution are both very simple. We do have other examples out here in the Pacific Northwest where the challenge is that we are seeing a lack of reproduction and we don't know that reason. So there was a study in Northern California by a researcher, Jeanette Howard, and she compared the size classes and age estimates of mussels between two different rivers. And what she found was in one, there was kind of a normal age size class distribution because, our mussels really can reproduce pretty much annually or probably, every other year or a couple years throughout their lifespan. And in this case, one stream had this totally normal distribution, but when she compared that to another stream in a different watershed, found that those mussels had not reproduced, or at least had not successfully recruited. Those young mussels were not making it for whatever reason. And so for 30 years, she saw this gap in mussels being part of the population. So, you know, in that case, there was no easy answer for why that's happening. And we do hear about that in other places in the Willamette. There was a population that was studied, and they weren't finding young Western pearl shell. So, you know, it's hard to know if that could be a function of water quality, poor habitat, lack of host fish. You know, and one other thing that we kind of alluded to, but that microhabitat stability is also really important for mussels. Those juveniles, when they fall off of the fish, they're the size of a grain of sand, and so they're really easy to be dislodged from their habitat. So if the fish aren't in a place where those juveniles can thrive when they drop off, that can really impact their ability to establish. So it's possible that you know, our activities with dam management, where we are changing the normal flows and velocity of water, that could have an impact. But yeah, it's so hard to know. I think we constantly wish that we could just ask mussels these questions. It'd be much easier.
Yeah.
So I feel like I probably know the answer to this, but it's always worth asking. I mean, they live a pretty long time, so they seem like they would be pretty durable. But like, if I'm tromping around in a stream bed, can I hurt anything?
Yeah, you can. They're usually pretty tough, and like us walking around, you know, there's sometimes some casualties. Really, something that can cause some issues is cattle access. We've seen a lot of cases where cattle's in the stream can really mess up a mussel bed, crunching a bunch of them, unfortunately. But as far as your presence in there, if you know that they're there, then you know, go horizontal as soon as you can, I guess. If you're trying to snorkel, that is.
And pull up my iNaturalist app.
Yeah.
And okay, here we go. Your comment on dam management leads me to ask about the impact on mussels in the Santiam downstream of the green peter release last year. Was that studied?
Yeah, and just for clarification, I think I know this, but the green peter release, was that where they actually did like a reservoir flush? Is that what happened?
Oh, I'm not sure.
Okay. Yeah, so no, I don't believe anything was studied there. We do know that mussels, we do know that they are impacted by things like sediment when it's released. If it settles, that can cause problems for them, similar to the debris flow that I showed that picture. When there are high flows, if they're in habitat that allows them to sort of maintain their position in the stream and they don't get scoured out, you know, they're able to sort of hunker down. And, you know, the joke is that they clam up. And so they can close their shell, they can sort of wiggle a little deeper down potentially to try to maintain their position when these flows change, because they are really, I mean, they evolved in rivers that are highly dynamic. The challenge is when we have less of the habitat in the stream that they had before. So the worse the habitat is, the less that they are able to, you know, do what they do in normal circumstances. So I'm a little less familiar with South Santiam. I haven't done many surveys in that area, so I'm not sure what would have happened. But when there were really high flows, gosh, I want to say it was maybe 2018 or 2019 in the Willamette, that caused a lot of flooding. Many people reported seeing just shells of Western pearl shell that had been scoured out of the stream and were found on the floodplain. They had just literally been scooped out of the river and left high and dry. So we know that there can be impacts when we have these dramatic changes in the rivers, but yeah, I'm not aware of anything with that particular instance.
Our questioner clarified that the drawdown was to encourage salmon fry coming downstream.
Okay. Yeah. I mean, mussels are adapted to, you know, fluctuating stream conditions. The missing piece is the good habitat. So.
Cool, thanks.
Yeah.
So another one from me. There's a lot of. Do you want to go ahead?
Well, no, I just related to the need for good habitat, and because our theme for the past couple of years have been beaver in the basin. I'm wondering, is there a connection or beaver any studies done on whether beaver created wetlands or beaver activity in general is good for mussels?
Yeah, beavers are a complicated one. Jack, are you familiar with any studies back east about beavers and mussels?
No, I'm not. The beaver mussel relationships is kind of new for me out here. Yeah.
Oh,
Yeah,
maybe
yeah,
future.
we have seen you know, one of our study sites is a Murderers Creek in the South Fork John Day Basin, and that's an area with a lot of beaver activity, with sort of elevated water levels and a little bit of slowdown in a dynamic creek, and the mussels there seem to handle that quite well. I have seen in other instances out here where a beaver dam has been built in an area that had mussels before, and that really sudden change to the immediate area around the dam did, you know, the mussels were not there later. So I think it depends a little bit on the stream itself and what kind of habitat remains after the dam, but it doesn't totally seem to exclude them. We see thousands of them in this other stream that has quite a lot of beaver, but I have seen them sort of eliminated from the immediate area around the dam, just because it does become so much deeper, and much more fine sediment. Yeah, but you know, the mussels, they evolved again in these streams that had a lot of beaver activity, and so I think you know, beavers do a lot of things that benefit the habitat that mussels do need, like slowing the water down so they don't get scoured out, and helping with that accumulation of sediment that can ensure that it's not just bedrock, because mussels can't live in bedrock areas. So yeah, so they benefit, but maybe also get a little impacted. It's complicated. You know, it's an ecosystem.
Right. Yeah.
Yeah,
Thank you.
Nature's always complicated.
always complicated.
So hot topic lately, climate-based migration. Are you seeing any of that in mussels and their host fish?
Oh, that's an
In an
interesting question.
interesting way?
Um, you know, gosh,
Yeah.
I'm not familiar with any positives from climate change for mussels. You know, there are so many interesting factors that I've read about how this like large scale change can affect them. And you know, an interesting one was a study that showed that it really can lead to a mismatch in host fish presence where mussels occur. So it actually can really kind of throw a wrench in the works. That was a study that showed that these mussels occurred in tributaries, and their host fish spent much more time in the mainstem river when the water got warmer, and so they really had less interaction. So I think probably that's more common where there are negative impacts. It's so tricky because our river systems are both highly connected and also, you know, very kind of in place. So as you know, species back east, like the ones Jack works with, might have been found in several river basins, but those basins don't, you know, it's like hundreds of miles to get from, you know, one location to another through the river network. So yeah. And Jack, your study in Tennessee, you were seeing differences in mussel abundances and species composition depending on whether you were sort of lower in the watershed versus higher, right?
Yeah, so in the downstream areas, there was a lot more diversity and abundance in the downstream areas where was generally more productive and warmer, and then the upstream areas where with the colder waters, there was less diversity and less abundances. And the theories behind why that was going on is for a number of reasons, but yeah.
Huh.
Yeah, one neat thing, just sort of related to the migration aspect, is that we often will find Western pearl shell in particular above fish barriers. And that's, it's so interesting to think about those relationships with host fish, because in those cases, there are generally resident trout, not salmonids. But, I've always been curious how related the mussels above, say, the waterfall is to the mussels below the waterfall, because sometimes those waterfalls have been around for a very long time. So another cool study, if anyone has a kid who's thinking about a graduate project, we've got a whole list for you.
Yeah.
Looks like we have a new question up if we have time.
Yep, absolutely. Yeah.
Do you know if any of the studies reconstructing historical population trends incorporate the effects of streambed dredging from the pre-Otto River navigation era? Pre-Otto River navigation era. Sorry, that was hard to read.
Yeah, I know from sort of the eastern US, there's more information on kind of what's happened post dredging. I don't know, Jack, if you want to speak to those declines.
I think the information is generally lacking. There's some areas where they have a little bit of information, but as far as like the the effort into essentially counting mussels and rivers and streams in the eastern US was like it spiked in like the early 1900s and then like died off for a little while and kind of came back again in the 80s. So there's like kind of a gap there when everything was going on. And so I think it's relatively unknown just because of the lack of information.
Yeah, and probably also, there were a lot of competing factors too. We didn't really talk about other human use of mussels, but in the eastern US, they were harvested, more than 1 billion were harvested for use as buttons. They would punch buttons out of a mussel shell. So they would collect, so many mussels, put them on barges and pull them out of the river. So it's probably a little hard to disentangle the impact of mussel harvest for that versus habitat destruction. And then also mussels can produce pearls. And so they have been used for a pearl industry, but generally the quality of the pearls that would come out of a wild mussel was quite poor. So if you buy freshwater pearls now, they actually come from populations generally in China, where those mussels are really raised to produce pearls. So the pearl industry is largely gone from North America. But yeah, their mussels were facing kind of everything at once in those early 1900s, from harvest to habitat destruction, and including dredging and dam building. So unfortunately, knowing what we've lost can be really hard. Yeah.
Wow. Well, Maya, did you have any more questions?
Just one,
Okay.
and it's another weird one, but then I'm done, I swear. So you said a couple of different things at different times that I'm kind of putting together into one thought. So you said that they take in and filter pollutants, which I'm guessing they mostly store in their tissues. But you also said that things that they don't eat, they kind of spit out for other things to eat. And I'm just wondering how, like, both either or, what happens to pollutants that they're sort of pulling out of the water.
Yeah, I can say something first, and then Jack, if you want to jump in.
Yeah.
Just with regards to what I was mentioning. Mussels will store, depending on the particular pollutant, they may store it in their shell. So in the case of DDT, one of the the breakdown of DDT results in different chemicals, and they actually have the ability to incorporate that into their shell material. So you can actually look at concentrations of DDT historically if you were to analyze a mussel shell. other pollutants like E. coli, they actually eat, so they completely break something like that down. And then other things they can store in their tissues. And so, yeah, there are several different pathways. The things that they tend to spit out, to my knowledge, are larger items that they wouldn't feed on necessarily. But knowing
Microplastics kind of thing,
what's that?
like a microplastics situation, or
Yeah, microplastics is an interesting one. You know, those things are so small that I think some of that can pass through and end up in tissue. There was a study at PSU a few years ago looking at microplastics in oysters, and they were finding that they did have microplastics in them. Yeah, Jack, I don't know if you had something to add to.
Yeah, I just wanted to add about, as far as the mussel diet, we haven't really talked about, what they eat exactly. And, generally we know that they, feed on algae and, organic material within the water column. But as far as like specifically what they're eating, the genre of phytoplankton, it's kind of unknown at this point. As far as, back to your question of picking which ones to filter out and which ones to not, that's kind of dependent, probably like Emily's saying, on the size of things, and certain types of algae, are in different size categories, so they might be picking the things that are the appropriate size that they might typically eat rather than the larger stuff, like Emily said. So yeah.
well, thanks.
Jack is going to be
Oh.
presenting at the Marys River Watershed Council Thursday, March 21st. So if you want a chance, I think you're going to bring some mussel shells with you and
Yeah,
get a
which I
little
have
bit
some,
more interactive.
I have some here, by the way. They're kind of hard to see. I
Oh,
meant
wow,
to
that
show
looks
these
big when
earlier.
you hold it up.
Yeah. This is a floater, the species is.
Nice.
Yep.
Yeah.
Awesome.
So it'll be another neat opportunity to hear some of the same information, but also little
Yeah.
different geography and some other cool stories we'll share. So
Yeah, and I'll
Yeah,
bring
and
some shit. So no, go ahead.
no, I was going to say, if there's a question that pops up, later on this evening, like, oh, I should have asked that, or, I wish I had asked that. If you're local enough, you can come on down and ask that question in person.
Yeah. And I'll bring some shells with me then for people to look at and handle, and some of the equipment we use in the field, and talk about that a little bit. So come check it out.
If it's rainy enough, are we all going snorkeling too?
bring your snorkel. You might need to with this rain. Yeah.
Well, thank you both so much, Jack and Emily. This was really fascinating, and they truly are weird and wonderful, aren't they? Those mussels. And thank you so much, Maya, for your help with the Q&A, and thank you so much to all of you who joined us tonight. Once again, like, if you would like to become or learn more about being a friend of the LWC, there's the link up there that Maya posted in the chat. And also, if you'd just like to sign up for our newsletter and get immediate emails when events come up, there's a link now to subscribe in the chat that Maya just put in. And I will look forward to seeing you at some future virtual events, but as well as in person as our weather continues to improve for spring and summer. So thank you all, everyone. Good night, and have a wonderful rest of your week.